Hello and welcome to our video lecture on section 18.2, where we will discuss major histocompatibility complexes and our antigen presenting cells. Now, these major histocompatibility complexes are going to be referred to as MHC molecules, and they're going to be expressed on the surface of our healthy cells, which helps identify them as being of our own body, of being a normal body cell, almost like an ID, like I belong to so-and-so's body. And it's presenting these to many of our immune cells. And they're going to play an important role in presenting any foreign antigens if we have a pathogen within our body, which can then help to activate our T cells. Let's go through our section learning objectives. We want to be able to identify cells that express MHC1 and or MHC2 molecules and describe the structures and cellular location of the MHC1 and 2 molecules. We also want to identify the cells that are antigen presenting cells and describe the process of antigen processing and presentation with MHC1 and 2 molecules. So let's first dive into our MHC molecules. These are made up by a collection of genes that code for them on the surface of all nucleated body cells. So hopefully right now you're thinking of all the different cells that we have within our body, and this would exclude our mature red blood cells that lack a nucleus. So they're not able to express MHC molecules on their surface. And when we're talking about our MHC molecules, do know that in humans we call these human leukocyte antigen genes, although we will be talking about them mostly as MHC molecules. And we've got two different types, and they're both going to be transmembrane glycoproteins, so meaning that they're going to kind of go through the plasma membrane here. And we could see their structures are a little bit different when we look at class 1 versus class 2. Our class 1 are on all of our nucleated cells, and on our MHC molecules, you could see that we have this antigen binding clef. And so on our class 1, we're able to display having our normal self antigens, identifying them as our own body cells, but we're also able to put out here our non-self pathogens or pathogen antigens to our effector T cells to let them know that our cell is abnormal or we've been infected with something like a virus. Whereas our class 2 or type 2 molecules are only found on our antigen presenting cells. So that would include our macrophages, dendritic cells, and B cells. So they're going to present abnormal or non-self pathogens as well in order to activate our T cells. So let's talk about those antigen presenting cells, or APCs as I'll refer to them. And they are made up of our dendritic cells, macrophages, and B cells. Now, as a reminder, only our MHC class 1 molecules are in all of our nucleated cells. So even these APCs will be able to have our class 1 MHC molecules, but it's these APC cells that are able to display the MHC class 2 molecules with an antigen in order to activate our T cells. Now there is a little bit of differentiation here. So our dendritic cells and our macrophages recognize pathogens through non-specific receptor interactions, so by utilizing those PAMPs, whereas our B cells can interact with foreign pathogens or free antigens by using their IgD or IgM receptors here that are able to bind to an antigen, and then the B cell will internalize that antigen by endocytosis. So a little bit more on this in the next coming slides. So let's take a look at how these APCs go about presenting an antigen with their MHC2 molecules. So first we're going to look at specifically the macrophages and dendritic cells, since it is a little bit different. They are going to process the antigens in order to display them. So they take up the pathogen just like through the process of phagocytosis, and then they're going to start to degrade the pathogen for antigen processing. And through that processing system, they're going to look for the most antigenic immunodominant epitopes, and they will place that on their antigen binding cleft on that MHC2 molecule so that they can present it to the T cell. Now, what about all of our other nucleated cells that only have MHC class 1 molecules? Well, when we have a normal cell that is healthy, we are going to constantly be degrading proteins. And what we'll do is we'll take that protein and display it on the MHC class 1 molecule. And as our immune cells, such as our NK cells, kind of come around and check our body cells, it'll recognize our self-antigen and there will be no destruction that takes place. However, if one of those normal cells are infected with an intracellular pathogen, such as a virus, then it takes that viral protein antigen that is specific to that virus and break it down utilizing its proteasomes, which is going to be kind of like an enzyme that breaks off the important antigen, protein antigen of that virus, and bind it with an MHC class 1 molecule, and it moves it out to the surface of the cell. Now, we have a goal of activating our immune system, and we do this through cross-presentation. And so we want that antigen to be presented on the MHC class 1 molecule on our unhealthy cell, which can be an antigen presenting cell, for our CD8 cells to become activated. And CD8 is just another way of referencing our killer T cells or our cytotoxic T cells that we're going to talk about more in the coming sections.